Okay, let me share my screen now. Okay, let us have a quick revision on what we have done in our previous lecture. In our previous lecture, I introduced you with the concept of structural alignment, structural superimposition, and how do we calculate RMSD. And I have also demonstrated you on how do we calculate this RMSD, how do we perform structural superimposition and structural alignments, as well as we have discussed some theoretical aspect of calculating these RMSD and performing this structural analysis. Okay, I hope I am clear. Okay, everything is visible as well as I am audible as well.
Yes,
Yes, sir.
you
Okay,
are.
okay. Well, now let us move on
Yes, sir.
towards application of this RMSD and structural alignments. Okay, so as I have already told you that we are going to discuss this application of structural superimposition and RMSDs using three basic examples. Okay, in first example, we would learn about how do we determine the functional relationship between proteins. For that, we would try to determine the relationship among hemoglobin, myoglobin, and leg hemoglobin as a typical example. Our focus would be like how these protein might have evolved, what might be the genetic event that might happen in the background so that these proteins are like as they appear today. Okay, so and we are going to use data that will be obtained from structural alignment and RMSD. Okay, next what we are going to do is that we are going to see how this RMSD is applied in drug discovery, specifically in validation of docking protocol. And finally, we are going to see how this RMSD can be used in molecular dynamics simulations as such. Okay, so this is what we are going to do in today's and the next lecture. Okay, so let us begin. Of course, I have also introduced with some key concept like what are homologs, what are orthologs, what are paralogs, and we are going to revisit uh, these terms today as well as in our next lecture when we deal with homology modeling. Okay, so let us begin discussing with the very first aspect of application of RMSD in determining the functional relationship between proteins. Okay, and our test proteins are hemoglobin and myoglobin. These two proteins are usually found in mammals, while leg hemoglobin is usually found in plants. So before we go on discussing how these proteins are related to each other, well, I would first like to give you some basic idea about some sequence analysis that I have done on these three proteins. Okay, well, let us first start comparing these proteins on sequence level, and then we would go on to structural level. Okay, and these the table that you see over here is basically the output, or you can say the results that I have obtained when I compare this hemoglobin, myoglobin, and leg hemoglobin with each other. I have downloaded the sequence of hemoglobin, myoglobin, and leg hemoglobin, and I have tried to perform pairwise sequence alignment. Okay, so now what you can see over here, this is the typical output of pairwise alignment between hemoglobin and myoglobin. Okay, And you can see over here that the percent identity between hemoglobin and myoglobin is found to be 27%. So this would eventually mean that hemoglobin and myoglobin share only 27% identity, and wherever there are identical sequence between these two proteins, hemoglobin and myoglobin, it has been shown over here in this alignment figure. So one thing to note is that this hemoglobin and myoglobin appears to share 27% identity between the two proteins as such. On the other hand, when I tried comparing myoglobin with leg hemoglobin, I came to a conclusion that myoglobin got aligned with only 22% sequence of leg hemoglobin. Well, say for example, in case of hemoglobin and myoglobin, as you can see over here, the two sequences were aligned with each other from one end to another end, right? Both the sequences were aligned globally, we can say. That includes both the ends of the two sequences. On the other hand, that was not the case in case of myoglobin and leg hemoglobin. Okay, so what it says that myoglobin and leg hemoglobin were aligned only in their 22%. That means not entire sequence was aligned, but only 22% of a small stretch of the two sequences was aligned. Okay, and in that alignment as well, that whatever short region of these two sequences got aligned, in that short stretch as well, only 26% residues were found to be identical, right? So by this you might now imagine how diverse this myoglobin and leg hemoglobin sequence are. Okay, the first case is that it did not align the entire two sequence. Okay, the sequence alignment or pairwise sequence alignment tool failed to align end to end sequence of myoglobin and leg hemoglobin, or in other words I can say that global alignment of myoglobin and leg hemoglobin was not possible. Okay, so and it happened that only 22% of the two sequences were aligned. Okay, and in that 22%, only 26% of amino acid residues were found to be identical. So this indicate that there is a lot of difference in myoglobin and leg hemoglobin sequence. Okay, and when I tried to align hemoglobin and leg hemoglobin, well, I ended up finding out that there is no significant similarity between hemoglobin and leg hemoglobin. Okay, so this is original sequence, some elementary sequence analysis result that I wanted to share with you, and these are the conclusion that I drew when I tried to align the sequences of these three proteins. Okay, uh, from these elementary result, it would be clear that you can say that these sequences are not very similar so far the sequence comparison is concerned. Okay, now let us move on from sequence comparison to structure comparison. Okay, so in our previous lecture, I have demonstrated you that how do we align hemoglobin and myoglobin, and how do we subsequently calculate the root mean square deviation. If you remember yesterday in our previous lecture, we have concluded that when we try to align or when we try to superimpose this hemoglobin and myoglobin structures and subsequently calculate RMSD, we got the value of 2.06 as root mean square deviation value, considering the C alpha carbon atoms in both the structures. Okay, in the similar way, I have also tried aligning hemoglobin and leg hemoglobin, and ended up with identifying the root mean square deviation of 4.42 Armstrong. So far, the C alpha carbon atoms are concerned in both the structure. While in case of leg hemoglobin and myoglobin, the C alpha RMSD was found to be 2.28 Armstrong. You can just check it out over here. And I have used the same protocol that I have used yesterday. Okay. Well, now just compare that. If you try to compare the sequences, they were found to be very divergent. Okay, you cannot say based on only sequence similarity result that these proteins might be related. Okay, so you might have heard people saying that if at least two protein sequences or two gene sequences, if they show at least 30% of similarity, you can call them to be similar to each other. But the best case scenario that we end up when we compare hemoglobin, myoglobin and leg hemoglobin is that you have got a 27% best score, and that is for hemoglobin and myoglobin. So one would consider by elementary sequence analysis that there are very less chances that these proteins might be might be related to each other. But when you try comparing the structures, you can see the clear similarity so far the structure is concerned. Both, all of the three proteins, as you can see, share the common fold. Okay, you can see that most of the secondary structural element and tertiary structure, the fold that these proteins have is very similar. Okay, and hence we say that the structure of any protein is more conserved than its sequence, and the answer lies in its function. Okay, well, say for example, if a protein needs to perform certain function, it is necessary that it needs to retain a particular structure. Okay, so unless and until the structure is maintained, it cannot perform some function. Okay, so that is why we say that although these three proteins appears to be sequentially divergent, or if the sequence appear to be sequentially diverse, but structurally these three proteins are very similar to each other. And the reason that you can say of having such conserved structure is that these three proteins need to perform similar functions. Okay, since it is a globin gene, well, these all of these three proteins have to do something with oxygen physiology. Okay, and hence the function remain conserved so far these three proteins are conserved. And in order to maintain the function, it is necessary that these proteins maintain the structure at the very first place. Okay, unless and until the structure is maintained, the function cannot be maintained. Okay, so it doesn't really matter how many mutations has happened when these proteins evolved from a common ancestor, but so far the function is supposed to be maintained. It is necessary that these structures are uh, are supposed to be these structures are supposed to be remain conserved in order to maintain the function. Okay, so now based on this structural data as well as the RMSD data, now we can compare these RMSD values, and we can then draw a few conclusion, like what event might have happened that might give rise to these orthologs and paralogs as such. Okay, so well, say for example, if you now compare the RMSD value, then you can say that since the RMSD value between hemoglobin and myoglobin is comparatively the lowest, you can say that this hemoglobin is structurally more similar to myoglobin as compared to hemoglobin, leg hemoglobin. Sorry. right? So based on this conclusion, based on this data, we can say that these paralogs, that is hemoglobin and myoglobin, are more closely related to each other. And since they are more closely related to each other, that gives you a kind of idea that these two uh, these two paralogs might share a common ancestor. Okay, this would mean that such a gene duplication event that gave rise to this hemoglobin and myoglobin, such gene duplication event might have happened later as compared to the speciation event. Okay, so this leg hemoglobin gene might have diverged way too earlier than the ancestor of hemoglobin and myoglobin. So what we can say, that the speciation event that gave rise to mammals and these plants might have happened at the very earlier stages in evolution as compared to gene duplication and mutation events that gave rise to paralogs like hemoglobin and myoglobin in mammals. Okay, so how do we come to draw this phylogenetic tree? We came to this conclusion solely on basis of root mean square deviation and the values that we have obtained from structural alignment. Okay, so you can use such data in determining how these proteins might have evolved, how these proteins might be related to each other, which genetic or evolutionary event might have happened earlier, or which genetic or evolutionary event might have happened later in evolutionary timescales. Okay, such kind of inferences can be made using this RMSD and structural analysis data. Okay, so this is one of the application of RMSD and structural superimposition in sequence as well as structure analysis as such. Okay, good. So now let us move on towards discussing what are how can we use this RMSD as a tool in molecular docking. So before I proceed towards discussing what this discussing the application of RMSD in molecular docking, well, I think it would be better if I first introduce you with the concept of molecular docking. Okay, so well, let us first talk about what is molecular docking. Okay, and then we would talk about the application of RMSD in molecular docking. Okay, now this molecular docking, you can define molecular docking as a technique that is used to predict if a certain ligand molecule will go and bind to certain protein molecule. Okay, well, now say for example, let us begin our discussion that this might be your protein molecule. Okay, and we usually call any protein molecule, when we talk about molecular docking, the protein molecule is usually referred to as target. Okay, and this target now can be any macromolecule. It can be protein, or even it can be DNA. Okay, because there are a lot of small molecules which are known to impart its therapeutic effect by directly binding to DNA. So when we talk about this molecular docking, the target can refer to any macromolecule. It can be protein or DNA. If it is protein, it can be anything, like it can be an enzyme, it can be a cell surface receptor, it can be it can also be an ion channel. Okay, so whatever protein targets are, they can be either enzymes or any cell surface receptor or ion channel. So wherever your small molecule is supposed to go and bind, it is called as target, so far molecular docking is concerned. Okay, so now you know what are targets. Okay, so now well, say for example, the purpose of molecular docking is to computationally predict if you know that you have a ligand, if this ligand would go and fit into the binding site of any protein. Okay, if it can bind, so if it binds to my given target molecule, how would that protein ligand complex would look like? Okay, that prediction is usually carried out using molecular docking. Okay, well, say for example, if you are working with say any enzyme, okay, in your project work, so you might be working with any enzyme. It can be dihydrofolate reductase, it can be any kinase that might be involved, or it can be any G protein coupled receptor. Okay, so those are called as protein targets. Okay, and what usually you do is that if you are a medicinal chemist, you might have developed a series of certain chemical molecules. Okay, this can be molecule 1, this can be molecule 2, this can be ligand 3, and then you would be interested to find out if whatever ligands that I have synthesized in my lab, whether these ligands have affinity towards any given target protein. Okay, and when you try to perform such a study in which you try to fit multiple ligand one after another in the binding site of protein, we say that you are performing virtual screening. Virtual screening यानी क्या? It is a technique by which you try to fit a series of ligands into the active site or binding site of any protein target molecule and test if they bind there or not. Okay, that is the sole purpose of this molecular docking. Okay, so let me first uh, show you how these things might look like when you perform molecular docking. Okay, so it's demonstration time now. Okay, so this here we have got a tool called as Python prescription, or what we call it as Pyrex. Okay, so this tool is used for setting up such molecular docking and virtual screening system and perform such molecular docking experiments. Okay, so this Python prescription is a graphical user interface for a program, a very popular program called as Autodoc. This Autodoc basically runs at the background, and what you see over here is just a GUI. So when you use this program, actually you are using that Autodoc to run your molecular docking simulations. Okay, so let me first show you how the results of molecular docking might look like. Okay, so what I have is I would try to load a target protein first so that it might give you a detailed description of what we are doing. Okay, so here we have in biotech in 2 and docking validation. Here is your protein molecule that I am loading. Okay, as you can see over here, this is a CPK representation in line model, and this molecule belongs to an enzyme. This model belongs to an enzyme called as cyclooxygenase 2, COX2, which is an effective target in anti-inflammatory response. Okay, so most of anti-inflammatory drugs that are present in market do target this cyclooxygenases too. Okay, so here is your protein molecule. Now I will load some results that we have already done, the objective of that study was to determine if the set of flavonoids can inhibit this cyclooxygenases or not. Okay, so what we have done is that I am showing you the results of docking this flavonoid called as diosmin into the binding site of the protein COX-2. Okay, so here we go. Okay, and as you can see, the ligand now appears in this stick representation. Okay, so this is the first pose of this ligand. Okay, then this ligand can orient in some different pose. This is second pose, this is third pose, this is fourth pose, this is fifth pose, sixth pose, seventh pose. Okay, eighth pose, ninth pose, 10th pose. Well, I have recorded 10 best poses that this ligand can orient itself into the active site of this target protein. Okay, and based on something called as this binding free energy, you can determine if these molecule can attach to this COX inhibitor or not. Okay, so this is how you might end up after performing molecular docking. Okay, these are the results. Okay, so what you see over here are typical results of molecular docking that are obtained when you try to fit any ligand into a protein molecule. Okay, is everyone with me so far so good?
Sir, what is this binding energy, unbound energy, internal energy, and all?
Well, these
And
are
why do we get different poses?
why do we get different poses? Well, that is what the objective of molecular docking is, friends. What are the different ways in which our ligand can go and attach to the binding site of this protein? Okay, so that is what our objective is. What might be the different way that this ligand molecule can orient itself into the binding site of protein? And so far this intermole energy, internal energy, torsional energy, and unbound energy is concerned. Well, as you can see over here, these are components of this binding free energy. Okay, and lower is the binding free energy, higher are the chances, or you can say greater is the affinity of that ligand to your protein molecule. Okay, well, let us not go into more details of that because that would deviate from our RMSD stuff. Okay, so my objective of showing this is that it is possible for you people to try and fit any ligand into the protein's binding site. Okay, यहाँ तक समझ में आ गया? Is it
Yes,
clear?
sir.
Okay, okay, ठीक है. So now what I will do is that I will remove this. Okay. Okay, now we will come to this thing a bit later. Okay, so now this is a software, right? This Autodoc or Python prescription, this is just software that eventually follows certain steps, a logic of steps that it is programmed to do for. Okay, do you agree with me with this thing? कि कोई भी रहो, molecular docking क्या है? It is a software. And what are software? Softwares are nothing but a set of commands that a programmer has designed. Okay, now if this is the case, how can you completely rely on the results that are obtained from such softwares. Why? Because it don't know the biology behind what is going on. Okay, then how do you make sure that if or not you want to rely on the results of this software? Do you get my point?
Yes, sir.
Okay, then how do we make sure that whatever results that this software is giving us, whether these results are biologically relevant or not, whether these results are biologically meaningful or not? बरोबर आहे? So these questions are needed to be answered first before you go on for testing your ligand molecules, whether it would go and bind to certain protein molecules or not. Okay, and in order to find out answer to this question, what you need to do is to first validate your docking system. That is what we say, that first you need to validate your docking system in order to make sure that whatever docking results that you get, those results are biologically meaningful, those results are scientifically relevant. Okay, so how do we do this validation now? Okay, so let me tell you what is the basic logic behind performing such validation study. Okay, so when you perform such validation study, and usually validation studies are conducted before you dock your test ligands. Okay, first you validate your docking system and then you dock your test ligand into say certain protein. Okay, so first let us talk about what is validation and how do we do this validation stuff. Okay, so to begin with, usually it is advised that when you perform molecular docking, you select a target protein that is co-crystallized with its inhibitor or say some substrate, if that is any competitive inhibitor if you are dealing with. Okay, so while you download the target structure from PDB, try making sure that you have certain structure of your target protein that is already co-crystallized with say some known inhibitor. Okay, so usually it happens in PDB that you end up with models that are co-crystallized with inhibitor. Okay, and that is very important so far your validation of the docking system is concerned. So once you have your target protein co-crystallize with any known inhibitor, what you do next is that you then separate your protein molecule and your ligand molecule in two separate files. Okay, and after separating, what you do is that you redock the same pose into the active site of your protein. Okay, बात समझ में आ रही? And after doing this, after redocking your co-crystallized molecule into your protein binding site, what are you doing? You are trying to test if your software gives the similar result as that of this experimental structure that you already know. Okay, so all in all, what you do is that you test the ability of any docking software to reproduce the experimental pose that is already known. Okay, now if your docking protocol or if your docking algorithm is successfully able to reproduce the experimental pose, that means you can rely on whatever values you get for your test ligand. Okay, do you go with this concept now? Okay, I will show you how these things work. Okay, the things would be very clear then. Okay, well, now say for example, let us stick on to the same case of COX and flavonoid inhibitor. Okay, so well, say for example, when I begin this study, I figured out that I have a model that is deposited in PDB that is already co-crystallized with the inhibitor. Okay, as you can see over here, I am showing this COX-2 molecule in ribbon. Okay, and I know that I have this naproxen that is co-crystallized with the inhibitor as such. Okay, okay, do you see here? Okay, here we go. Display, so 3D. Okay, so here whatever you see in this stick representation is your inhibitor naproxen, this one that is in complex with your COX-2 molecule. Okay, one more thing that you need to understand is that if your naproxen molecule or your inhibitor molecule orient itself in this particular pose, then and then it would go and make certain polar interaction or whatever interaction it is supposed to make in the binding site of this protein, and stabilize this protein inhibitor complex, and eventually result in inhibition of this protein. Okay, बात समझ में आ रही है क्या आपको? So this pose of this inhibitor, we call it as experimental pose. Why? Because it is determined using X-ray crystallography, and we are sure in this case that if my ligand orients in this way, this particular way, it would result in you can say successful inhibition, or you can say it would result in productive inhibition. Okay, do you agree with this thing?
Yes, sir.
Okay, so now what I will do is that I would kind of separate these two molecules first. Okay, after separating, I have already done it. This is the NPS molecule, and this is the apoform of we would call it as apoform of your COX. Okay, so once I have separated these two molecules, then I would perform that molecular docking. Okay, and after performing molecular docking, I would get some results that I'm going to show you. Okay, well, now let me load this apoform of your COX molecule. Okay, now I will load the results that I obtained after redocking the separated inhibitor back into the active site of your protein, or your target protein as such. Okay, now you say that there are different orientation that this software has predicted over here. So this is the first orientation. Your ligand can orient in this way. Now you can see that there are certain changes in the functional group. It can orient this way, it can orient this way, it can orient this way. There are multiple orientations in which there are multiple orientation that your ligand can take up in the active site of this protein. Okay, so these are the predicted results. For time being, to make things more clear, I would now hide the protein chain. Okay, so that you can get key, okay, this is happening in the active site. Okay, but we don't need this protein molecule for our further discussion. Okay, so these are what? These are predicted results. Okay, this is first pose, this is second pose, third pose, fifth pose, sixth pose, seventh pose, eighth pose, ninth pose, and 10th pose. Okay, and every pose is associated with say certain binding free energy. Okay, so this pose has got binding free energy of minus 8.67. The next pose has got binding free energy of minus 8.85. Okay, so now what we do is that, well, let us try to compare how our software has predicted the poses as compared to the crystallographic pose, right? In order to do that, I will load the crystallographic pose now. Okay, so here is, now I am loading the crystallographic pose. Okay, so whatever is moving is the predicted pose, and this one is the crystallographic pose. This one is predicted pose, and this one is the crystallographic pose. Now let us try and structurally visualize how our software has performed. Okay, so as you can see, the very first pose might not appear to be very similar to the experimental pose or crystallographic pose as such. Okay, but when we move on to next result, you can see that this pose is almost identical, or it is almost similar to the crystallographic pose, right? There is only slight difference. दिख रहा है यहाँ पर? Okay, and fortunately this pose, which is highly similar to the crystallographic pose, end up to be having the best binding free energy. Lower is the binding free energy, higher is the affinity. So what do these results say? That this particular second number pose, which is having the highest binding free energy, is very similar to your experimental pose, right? And hence you would use the same setting for molecular docking as you have used for your molecular docking, right? Well, whatever setting that you have used to perform this validation experiment, the same setting you would use for performing virtual screening for your test ligand as well, so that it would give you biologically meaningful results. Do you get this thing? Are you guys still with me?
Yes, sir.
Okay, so now how do we compute that whatever pose that we have predicted is similar or dissimilar? There should be certain measure, and that measure is nothing but your RMSD. Okay, so by comparing these structures of these poses with the experimental pose, what you would do is that you would try to determine the RMSD between the experimental pose and first pose, calculate RMSD between experimental pose and second pose, calculate RMSD between experimental pose and third predicted pose. Okay, so in this way you can compute the RMSD and then determine if the predicted pose is similar to my experimental pose or not. Okay, and if the RMSD value is low, you can say that your validation protocol is successful. That is, your software has successfully predicted the experimental pose, or it was successful in reproducing the experimental pose as such. Okay, so you don't have to calculate this experiment, the RMSD value between experimental pose and your predicted pose. Okay, your docking result itself would have this data. Okay, so this is the docking log file that you usually end up after performing this validation experiment. If I open it in any text editor, okay, and if I search for terms like RMSD, okay, you might end up in this table. Okay, so over here it says this reference RMSD is the table that you might be interested in. So what the values in this table says what is the RMSD between this particular predicted pose and the experimental pose, or what we call it as the reference pose. Okay, so now scientific community has came to a kind of consensus that if your software is able to predict a pose whose RMSD is below 2 Armstrong as compared to the experimental pose, you can say that it is successful in generating the in validating your system. Okay, so you can say that your system is validated if the RMSD between the ligand molecule, or you can say the predicted pose and the experimental pose, is less than 2 Armstrong. Okay, is it clear? So now if 2 Armstrong is the cutoff of determining if the software is able to regenerate the pose or not, then the result that we have obtained here are way too more significant, right? The best result that we have obtained has got the RMSD of only 0.34 Armstrong, right? And in this way you can conclude that okay, we were able to successfully validate our protein ligand system or our docking system, and hence we can now use our test ligand to determine if they can go and dock into the binding site of protein or not. Okay, and here my friend is the application of RMSD in molecular docking. Okay.
Yes, sir.
Sir,
हाँ बोलो.
sir, on what basis this cutoff value is set at 2
Yes,
Armstrong?
yes, 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 yes. That is. I was expecting this question, but you would have to wait for a while. Just hold on to that thought till our next few discussion. We will come to this point.
Sure, sir.
Okay, I'm very happy that someone rose this question. Okay, good. तो इतना समझ में आ गया सब लोगों को कि what is the application of RMSD in molecular docking, where and how this RMSD concept is implemented. Now you also know how you can implement the concept of RMSD determining the in determining the functional relationship between the proteins. Okay, so these are just a few applications. Okay, so I think we would stop here today. Why? Because the next topic that we are going to deal with, that is application of this RMSD in molecular dynamics and convergence test, that would take some more time. Okay, so only 4 or 5 minutes are left for your lecture to get over. Okay, so we would continue our application of RMSD discussion in our next session. Okay, so well, discussing this RMSD and RMSF is just a uh, uh, just an excuse to get you introduced with the concept of molecular docking and molecular dynamics. Okay, because in your syllabus I don't think you have got these topics. Okay, and these are hot topics so far the computational biology is concerned. Okay, so it took us a while to finish up with this, but I think it would be productive for you to understand this concept and utilize them in your further project as well. Okay, good.
Yes, sir.
So I am stopping this recording now.